Hey, Rose. Did I just see you? It disappeared. Okay, my Zoom pane disappeared. Oh, there it is. Okay, all right. Why? Why did it disappear? Hey, Rose.
Yeah.
What's that? I didn't catch that, Kelsey.
Unprepared.
Oh, unprepared. For reasons of you don't have as much data as you wanted, or just like, oh crap, it's coming up Monday and I haven't really prepared myself yet.
Mostly just haven't prepared myself. Well, I did just I just finished my last thing the other day. So I just finished my data analysis yesterday. So that didn't help, but mostly just unprepared because I have had a million other things to do. And so research has not been the top of the list because it's later on the due dates.
Yeah, of course. And Kelsey, you're the first senior student who just got their last thing done like days before the final presentation.
Oh, I'm sure.
Yeah, right. That's true. Yeah, no, you're in the same boat that so many other senior students have been. So once again, don't sweat that. You're unique in that you're just like everybody else.
Senioritis never fails.
Well, everything, this is one of the difficulties with academia is like, in theory, there aren't due dates, and like if you're a newspaper reporter, there's a deadline. You have to have your written thing into your editor by 3 o'clock on the day of, so they can get it typeset, so that they can get it printed in order to go out the next morning. Academia doesn't necessarily have those types of deadlines, and the thing about academia is that we can shift deadlines whenever we want to, and that's great because it gives you a great deal of flexibility. But then it's also not so great because nothing has a deadline and everything has a deadline, and it's difficult to stay on track and be productive in those kinds of situations. So anyway, all right. Only one person got back to me about what you wanted to talk about for the remainder of our time together, so I just went with interspecific interactions because that was kind of on the plan anyway, and I think it kind of factors into the last thing that I want to talk about, which is conservation issues. Because as it turns out, at least for amphibians and reptiles, there are a number of conservation issues that are directly related to interactions of amphibians and reptiles with other species. And in the case that I'm thinking of, it's disease-causing organisms. And so that's what we'll do for today. And then into Monday, and then into Monday, we'll start talking about conservation more specifically. Next Friday, there's a paper that has been on Moodle for a little while now. It's in the last week. It's a paper by Miller, and it's looking at trends of, if I'm recalling correctly, reptile and amphibian decline in the United States in a geographic context, using a set of long-term monitoring stations around the United States that have been following populations of amphibians and reptiles for literally decades now. One of my office mates from when I was a master's student is one of the co-authors on the paper, and it's an interesting paper from the standpoint of conservation, and that will have a literature summary associated with it. So on Friday, come to class with your literature summary turned in, and be prepared for any of you to be called upon to lead discussion of the paper. Because it's a geographically explicit paper, there are some methods in there that you're not going to find familiar. Try and just work with it to the best that you can, simply because you've never been in a class where we've taught you the kinds of techniques that they're using in there. But I think it'll be intuitive enough that you can get through it without too much difficulty. It's a paper published in one of the Nature journals, so it's also structured in a strange way, because Nature structures their papers unlike a regular journal article. So that's on tap for Friday, and then that's it. We're done for this semester. So,
Is it possible for us to get our second lit sum back before then? I don't know where you're at with grading. Just wondering.
which lit sum was that?
Huey et al, something like that. I don't know. I don't remember. Anybody remember?
Oh, Buckley and Huey.
Yeah, that.
Oh, I had graded the Helmus ones, hadn't I?
Yeah.
I handed back Helmus to you, and then we did cover Buckley and Huey. Yeah, no, I need to get those. Yeah, that slid totally off my radar. Yeah, I will work on those this week and get them back to you on Monday.
Great, thank you.
Yeah, thanks for reminding me of that. I had kind of forgotten about that. I've been concentrating on other grading assignments. I didn't even think about the literature summary. Losing my mind. What else is new? All right. So interspecific interactions. The first thing that you want to be able to do with interspecific interactions is have a framework for remembering what the interspecific interactions are and what the consequences.
We can't see the slides if you're trying to share your screen. Just a heads up.
I thought I had. Let's see. All right, can you see? You can't see the slides now. Why is screen sharing not working? Can you see the slides now?
Yes.
Okay, all right. So the first thing you need to be able to do is communicate a framework for understanding what the different interspecific interactions are and what the effects on the different species interacting are. So these are competition, predation, parasitism, mutualism, disease. And one of the things I want to talk about as we go through this is how they kind of relate to species diversity and conservation issues. Within the area of diet, which organisms compete for access to food, they have foraging behaviors related to getting enough food, et cetera, et cetera. And so there's a lot of literature out there, especially in amphibians and reptiles, about the difference between dietary specialists and generalists and the factors that might determine whether you tend towards specialization or generalization. Looking at ontogenetic size, oftentimes we see that individuals' diets change as they get bigger once they're in their kind of final form, but we also see fundamental shifts in diet as, for example, larval amphibians go through the process of metamorphosis to become land-dwelling organisms. So their diet as larvae is very different than their diet as juveniles and adults. So you should have knowledge of all of these things. As well, there are some very specialized sense organs that are involved in the interactions of organisms. So in snakes, there's a particular organ called the vomeronasal organ or the Jacobson's organ, and that plays into prey capture, finding mates. And so we'll study that in some detail and how it operates. The idea of foraging modes, whether you're a sit and wait predator or whether you're a more active forager, is an idea that was developed by herpetologists and has become more widely used in the ecological community in terms of how one goes about finding food and capturing food. And so we'll talk some about foraging modes. Parasitism is an issue in amphibians and reptiles. Amphibians and reptiles are plagued by parasites, both internal parasites and external parasites, and that has consequences for them. Parasitism being another type of interspecific interaction. And then predation is a pretty important interspecific interaction. It varies taxonomically, and it also varies across life stages in groups with complex life cycles like amphibians, because the type of predator that you come into contact with in a larval environment might be different from the type of predator that you come into contact with in the terrestrial environment. And part of this is, of course, thinking about different ways in which amphibians and reptiles can avoid predation. And one of the conservation issues that we confront occasionally is the introduction of new predators into environments that didn't have those predators before. So for example, with bullfrogs that have been introduced, for example, in the Sierra Nevadas in California, there's this large-bodied aquatic terrestrial predator that lives in these high mountain lakes now, where those large-bodied predators used to not be, and they're having two effects on the native species that live there. One is they're competing for food in ways that competition hadn't been structured in those environments in the past. And the other way in which they're negatively affecting amphibians in those areas is just by eating them, consuming them, because it's a new predator. So all of these interspecific interactions have relevance to conservation issues. And so that's one of the reasons that I wanted to cover the information in this chapter, even though we've now skipped over some chapters that are perfectly interesting, like looking at mating systems, looking at communication systems in amphibians and reptiles. Those are interesting topics. It's just we have a certain amount of time that we can get to things, and I'm choosing this subset of information rather than some other subset because I want to get to conservation in the end. So if you were going to give somebody a framework for organizing different types of interspecific interactions, what would that framework look like? What would you, if you were explaining interspecific interactions to your mom or dad, how would you go about that process? Ideas. And we gave you a framework for doing this back in evolution ecology, but you probably don't remember it. But it'll probably start to come back to you when we get into it a little bit. How would you attempt explaining different interspecific interactions to your parents or grandparents?
Like there's competition between members of the same species for resources and food and shelter, but then there's also competition between members of different species for those same things.
Okay, so we're talking about interspecific interactions in this case. So let's just start with competition. So if we have competition, what's the nature of competition? What's going on in competition?
There's a limited amount of resources, so animals or whatever have to compete for those resources so that they can survive and continue reproducing or whatever and increase their fitness.
Okay, so species A and species B are competing for a common resource. As species A eats more of the resource or consumes more of the resource, what does that leave for species B?
They're going to be in trouble.
Yeah, and if species B is eating a resource or is consuming a resource that is useful for species A, what's the consequence for species A?
The opposite. Species A will be limited.
Yeah, yeah. So in competition, we can think about competition as having effects on species A and species B. And because these two species are competing for a common resource, as either species use it up, there's less available for the other species. But while the one species is consuming it and making less available for the other is consuming it, making less available for the first. And so this is an interaction that has negative effects on both species A and species B. In both of these cases, if you got rid of the other species, it would be better for the species that remains. And so competition can be defined as an interspecific interaction where the effect on species A is a negative effect and the effect on species B is a negative effect. So one framework for defining interspecific interactions is to think about what the effects of the different species are. What are some other interspecific interactions that we have to think about? Competition is certainly one of them.
Predation.
Okay, so predation is another type of interspecific interaction. Was that Rose?
It was Quinn.
Oh, it was Quinn. Quinn, what are the effects on the two species in predation?
Well, like species A would be the predator and then species B would be the prey. So, like species B would be like dying off more.
So that's a negative effect for species B. And what's the effect of the interaction for species A?
A positive effect.
Yeah, you're getting food, and the food happens to be the other species. So we can think of predator-prey interactions as an interspecific interaction that is positive for the predator and negative for the prey. Is there another interspecific interaction that is positive for one group and negative for another group?
Parasitism.
Is that Elizabeth?
Yeah.
Okay. And it is positive for the parasite, and it is negative for the host. Now, it's not negative in the same way that it is for a predator-prey interaction because it's in the best interest of a parasite to not kill its host. But what it does is it siphons off resources that the host could be using, and the parasite benefits as a result of that. There are a number of other important distinctions between predation and parasitism. In almost all cases, the predator is larger than the prey in terms of body size, whereas the parasite is in almost all cases much smaller than the host. So there are body size differences between these two things. There are differences in terms of what the ultimate negative consequence is to prey versus the negative consequence to host. But in terms of population modeling, if you were trying to model the effect of predation on a population in terms of its growth dynamics, that mathematical model for describing predator-prey dynamics is exactly the same as the model that you would use for modeling parasitism and its effect on the population dynamics of a host, for example. And it's because these things, these fundamentals are the same. It's positive interaction for one species and negative interaction for the other. what other kinds of interspecific interactions are there?
Mutualism.
Okay, mutualism. What are the consequences in this case? Is this Kelsey?
Kiara.
Oh, Kiara.
Well, if they have a mutualistic relationship, they're both benefiting.
Okay, so a mutualistic situation, a situation where there are positive effects for both species. It's pretty common to find mutualisms among plants and fungi and things like this, but it's harder sometimes to find this in the animal world. It is especially hard to find this in the herpetological world. One example of this is actually an interaction between owls and blind snakes. So in Texas, there was a guy named Fred Gelbach who noticed that owls were collecting blind snakes with these little thread snakes that feed on ants and termites primarily. And the owls were picking them up and carrying them back to their cavities. So owls are cavity nesters. And he would go up into the nest and look and see what was in the nest, and he would also look at the owl pellets. You guys know what an owl pellet is?
Owl poop.
It's not owl poop, it's owl regurgitation, but you were close, just the other end.
Still gross, still disgusting.
Yeah, so what happens when an owl eats animals, they swallow them whole, and after they have digested them, they will then puke up a pellet, and in the pellet are the fur and bones of the animals that it had eaten, so that it doesn't have to then pass them out through the cloaca, it just regurgitates them up. And it regurgitates them, and they collect at the base of the tree. So if you find a tree with an owl cavity in it, at the base of that tree, you will usually find a pile of owl pellets. And owl pellets are useful because then you can get a pretty good idea of what kind of vertebrate prey owls are eating, because the bones and fur of those animals are in the pellet. You guys know Ann Nickel? Do any of you know Ann Nickel? She used to teach E&E labs here. Anyway.
Yeah, she was still here our freshman year.
What's that?
She was still here our freshman year. We had her for E&E lab and then BD.
Okay, you had her for E&E lab. So she was a student here at Jewel, and her senior thesis involved looking at owl pellets from around Liberty and quantifying the prey of owls. For years, we still had her owl pellets up here. I think she finally took them home. Well, when Fred Gelbach was looking at the owl pellets at the base of the trees of these owls that he had seen bring blind snakes back to their cavities, he didn't find any vertebra of blind snakes. And so then he climbed the tree and looked in the cavity itself and found that inside the cavity, the blind snakes were living there happily. And he figured out over time that what the owls were doing is they were collecting these blind snakes, bringing them back to their cavity, leaving them in the cavity and not consuming them. And while they were there, the blind snakes were basically getting rid of all of the pests and parasites that could have a negative effect on the growing owlets that were living in the cavity. So they were basically feeding on these small invertebrates like ants and termites and parasitic bugs. And so that's a mutualism between owls and blind snakes. The blind snake gets a safe place to live. It gets a ready food supply, and the owl benefits from having fewer insects being pests to the developing owl babies. So it's unusual to see that in amphibians and reptiles, but you do find those things occasionally. Other interspecific interactions. And at this point, I'm thinking about the interaction between Mediterranean geckos and human beings, because I work on Mediterranean geckos.
Commensalism.
Thank you very much. What is the nature of commensalism?
It's positive for one species and neutral for the other.
It's positive for one species and neutral for the other species. So why is that the case for Mediterranean geckos?
Because they benefit, like they get to live on buildings and other architecture.
It's a place of shelter. It's also a place of potential food. And the presence of geckos doesn't have any discernible effect on humans, except when you occasionally go in and get your coffee cup out of your cabinet and there's a dead baby gecko in it because it fell into your coffee cup. And certain types of ceramic glazes are resistant to the van der Waals forces of the toe pads on geckos, and so they slide down into the coffee cup and can't get themselves back out. So these are the various types of interspecific interactions that we typically think of, and a useful framework for thinking of these is to think of them in terms of what are the net effects on the two species involved, and then classifying them on the basis of those net effects. Does this framework get at all of the nuanced details of each interspecific interaction? A simple yes or no question.
Can you repeat the question? Sorry.
Does this framework of thinking about interspecific interactions, is it particularly nuanced?
No.
No, it doesn't. It doesn't get into all the nitty-gritty details, but if you see an interspecific interaction and you just need to put it in a category, this is a decent way of figuring out what category that interaction might belong to. So if you were trying to get across to your parents or your grandparents what interspecific interactions were, this would be a useful framework for doing that. And so it's also useful for you to think about it in this way as well. So when we start thinking about interspecific interactions, one of the first things that we might think about are organisms eating other organisms. And so this will have to do with predator-prey interactions. It will have to do with competition. If one of the things that organisms are competing for is food. And so we can begin thinking about interspecific interactions in terms of what are the diets of amphibians and reptiles. And the diets of amphibians and reptiles are going to be affected by a lot of different things. Physiological constraints of the organism doing the feeding. What type of foraging mode does that individual employ? What type of sensory capabilities does that individual have? When is that individual active? Is it a diurnal organism or is it a nocturnal organism? Well, that affects what sensory capabilities it might use in finding food. You maybe use different sensory capabilities if you're nocturnal than if you were diurnal. Also, the time of activity affects thermoregulation. Among the conflicting activities that you have to balance, this idea of balancing conflicting demands, at the same time that you're gathering food and avoiding becoming food for something else, you also have to regulate your body temperature in such a way that you can function adequately. All sorts of biomechanic constraints in terms of the shape of the jaw and the head and the tongue, et cetera, that influence the kinds of things that you can eat, or are actually evolved responses to the kinds of things that you have evolved to eat over time. Body size influences how big prey items you can fit inside of your mouth. And then the resource availability affects what you might eat. You may prefer one particular type of food, but if the only food that you can get is this other type of food, that may be what you eat simply because you don't have access to your preferred food type. So all of these things can influence what becomes a part of the diet of any given organism. The other thing that I want you to recognize is that as you move from an egg to a larva to an adult frog, all of these things may change. The sensory capabilities that you have are different when you're an adult frog versus when you are a larva. Your time of activity is probably different. Certainly your location of activity is different. Your body size is different. Your foraging mode probably changes. So in organisms with complex life cycles, the diet should be expected to change in radical ways as the life form of the organism changes. So the rule in amphibians and reptiles is carnivory or insectivory, eating mammal flesh or other amphibian reptile flesh versus eating insects. And this is the rule rather than the exception. The exception, of course, is those organisms that eat plants. And so there are herbivores within the amphibians and reptiles, but that's the exception rather than the rule. Oftentimes, the size of the feeding structures is more related to the size of prey than it is to the size of the individual, and we'll see some examples of that today. There's also this dichotomy, although it is on a continuum between dietary specialists and dietary generalists. Generally, organisms that are insectivores are more generalist predators than organisms who are feeding on, for example, mammals or birds. But that's a general idea with plenty of caveats to it. But all species are to some degree or another selective. Even generalist species don't eat anything. They'll eat a lot of things within a broad spectrum of prey items, but they won't eat, every imaginable prey item. So when we think of dietary specialists, your book talks a lot about ant specialists, and I think it does it for a couple of reasons. Is that there are some common morphological adaptations that accompany ant and termite specialism. And there are actually just a number of diverse taxa within amphibians and reptiles that have settled on this particular form of dietary specialization. So to the left-hand side here, we have a narrow-mouthed frog. This is in the genus Dasterphrine. These are the microhylid frogs. And this is an ant and termite specialist that occurs here in Missouri. It also occurs south and west of here. This is the Mexican burrowing toad that lives down in the Rio Grande Valley of Texas and down into Mexico. Both of these species have pointed snouts, and both of these species are species that specialize on ants and termites. On the right-hand side, we have two species that you've seen before. We have Mullet horridus, the thorny devil from the Great Victorian Desert in Australia. And we have the horned lizard, which is found in Kansas, down into Oklahoma and Texas and further west. All of the horned lizards in the genus Phrynosoma are ant specialists. They feed on Pogonomyrmex harvester ants. And the thorny devil from Australia is also an ant termite specialist as well. In both of these cases, they have evolved convergent body forms that are associated with their ant eating proclivities. However, your book points out not all ant specialists take on this kind of short, squat body form with short snouts that are flat that allow you to eat ants. This is a skink. I had the genus in my head last night. Ray Huey published a whole paper on this species. Typhlosaurus. This is a Typhlosaurus. It's a legless skink from South Africa. This is a picture taken by a guy named Tyrone Ping. He is a very good photographer who specializes in photography of South African amphibians and reptiles. So this is a skink. It looks like a worm, but it's actually a skink. These are cycloid scales going down the body, making it look very shiny. This is also an ant specialist. The blind snake that I was referring to earlier is also an ant specialist that takes on a serpentine form because it's actually a type of snake. It actually is so much of an ant specialist that it mimics the pheromones of ants, so that when it enters an ant colony, it secretes pheromones that tell the ants that it's an ant, not a snake, and then it goes down into the nurseries where the larvae are found and basically consumes the developing ant larvae. So ant specialization is one type of dietary specialization. In some cases, it leads to convergent evolution of form, and but those aren't the only forms that this particular specialty can result in. Another example of dietary specialization mentioned in your book are the sea snakes in the Malay Peninsula. 16 species, only one is a generalist. All the others specialize in a particular way. Some of them specialize on eels, some specialize on different types of gobies. Catfish, one species in particular, there's a picture of it in your book, specializes on fish eggs. So there's quite a lot of dietary specialty out there in the world, especially within amphibians and reptiles, but there's also a lot of generalist sort of species out there as well. So virtually all of the lizards in the United States are dietary generalists. There are a few exceptions, but most of them are just generalist insectivores. The horned lizards being one exception to that. Another way in which there's kind of dietary specialization, if you want to think of this, is cannibalism. We've talked about cannibalism in the past with reference to spadefoot toads. So these are figures from a guy named David Finnig. He works at North Carolina State, I believe, and he works on cannibalism in amphibian groups, in particular the spadefoot toads of the American West. And here is a pond that is drying up, and all of these little lumpy things in this pond are spadefoot toad tadpoles. So these are these ephemeral pools that they dry up very quickly, and it's a race to get out of the pool, because if the pool dries up before you can metamorphose, you will die as a result of it. As it turns out, spadefoot toads start developing lungs even in the tadpole stage, so they can actually live for quite some time in a pond that is even this dry. But you'll notice that there are tiger beetles congregated around this pool of tadpoles. And so it's not just that you have to escape the pool before you die, you have to also escape the pool before terrestrial predators discover that you're there and easy to access and kind of vulnerable because you're not in a big body of water, and they come and begin feeding on you. Well, one of the ways to get out of this pond quickly is to just grow quickly by consuming your fellow spadefoot toad tadpoles. And so here we have a cannibalistic morph of a spadefoot toad feeding on a non-cannibalistic morph. This is a highly specialized form of predation. You are at this point a dietary specialist because you're basically eating members of your own species. So you have a monospecific diet at this point. What it enables you to do, though, is to grow much faster, reach the size required for metamorphosis much faster, and it allows you to emerge from the pond before the pond either dries up and kills you, or dries up enough that predators are attracted to it and you get killed by predation. Cannibalism also occurs in tiger salamanders, and so this is a figure from your book showing the morphology of a non-cannibalistic larva and a cannibalistic larva. It's weird because the way they've drawn this, it makes the brow ridge look almost like this is an angrier morph. But the main thing that you need to think about is this is a more sloping shovel nosed sort of morph than the rounder morph here. This is a morphology that actually translates into the metamorphosed organism, so that you can see adults have a different morphology depending on whether they were non-cannibalistic versus cannibalistic morphs as larvae. So cannibalism, another form of specialization. There can also be cannibalism on eggs. This is Eleutherodactylus coqui. Remember that it is an eleutherodactylid frog. They go through direct development. The female lays the eggs and the male guards the eggs. And there will be other males or females who will come by and try and eat the eggs because the eggs make a good meal. They're full of energy, and as a result, the male guarding the eggs will attempt to protect those eggs from egg predators. In certain parts of the tropics, there are snakes that specialize in eating amphibian eggs, and so you will see a lot of direct developing frogs throughout the New World tropics in particular, sitting and guarding eggs to keep those eggs safe from snake predators that specialize on amphibian eggs. Cat-eyed snakes, in particular in the genus Leptodyra, are particularly good at this. There's one species of Leptodyra that lives in the desert southwest of the United States. It does not specialize on eggs, but many members of the genus in the tropics do. I mentioned herbivory. So herbivory is the exception rather than the rule, but there are some common characteristics of herbivores within reptiles in particular. In all these cases, the act of herbivory is aided by microbial communities in the gut of these organisms. Most vertebrates lack the appropriate enzymes to break down plant material, and as a result, they usually have symbiotic interactions with microbial communities who do possess the enzymes to break down plant material. And so while you may be ingesting the plant material, it's really the bacteria in your gut that are digesting that plant material. As a result of this, you usually have to have a large gut in order to harbor these microbes, which means that you generally see herbivory occurring more in larger organisms. There may be other evolutionary reasons why larger organisms tend to be more herbivorous, simply because herbivory is a more energy efficient way of eating, so you can support more large organisms that are herbivores than you can large organisms that are carnivores. Outside of large carnivore or large herbivores like tortoises and the large herbivorous lizards in the family Iguanidae, many, if not most, anuran larvae are herbivores. They feed off of algae that are growing on the surfaces of leaves and rocks in ponds and streams. And rather than bacteria in their gut that do the digestion of the plant material, in most cases in anurans, in frog larvae, you have nematodes that live in the guts of tadpoles that digest the plant material. Interesting that even though you might see frogs and salamanders in the same pond, in that pond, frogs and salamanders are generally eating different things. Tadpoles tend more towards herbivory, whereas salamander larvae tend more towards insectivory, and so that's a fundamental difference there. Another form of herbivory is nectivory, which is the feeding on nectar. This is especially prevalent in day geckos in Madagascar in the genus Phelsuma. You will see these individuals sitting at blooming flowers, lapping nectar out of the nectaries of flowers. These geckos are found pretty commonly in the pet trade. There's some wild populations of them in Florida, and we had a day gecko that I collected in Florida back in 2012, who just recently died earlier this semester on us, and we would feed him nectar a couple of times a week, as well as some insects. As organisms grow, it's not surprising that their diet would change. So this is Nerodia erythrogaster. What is the common name of this snake? It's one of the species from lab. One of the common names is found in the scientific name. What does erythro mean?
It's a water snake, and that is literally all I know.
It is a water snake. Very good.
Learn the common names because we didn't have to know the common names.
Yeah, I know. I'm switching things up on you. What do you call a red blood cell? What's a scientific name for a red blood cell?
Erythrocyte.
Erythrocyte. So it's a red cell. Cyte is cell. So this is an erythrogaster. It's a red-bellied water snake. Oftentimes they're called plain-bellied because they're not always red-bellied. Sometimes they're kind of yellowish-bellied. But the red-bellied water snake, it's a species that occurs here in Missouri. And you can see from an analysis of the diets of different lengths of snake is as the snakes get bigger, they shift from a diet that is dominated by fish in young, small individuals to a diet that is dominated by amphibians in the larger animals. And this probably reflects an ontogenetic shift in where the individuals are feeding. Small snakes are probably feeding more in water and at the water's edge rather than up on land, probably because there are fewer predators in the water in the environments where these guys occur than there are up on land. As they get older, they can venture onto land more, and as a result of that, they go through this ontogenetic shift in the diet. So when you think about what an organism is eating, think about what's going on in the life of that organism, what size is the organism, where does the organism live at that time period. If you are living in a place like Cooley Lake, those are fishless ponds. And so if you're a small snake, the pond is actually quite safe because it lacks any large vertebrate predators like fish that might consume you, and it has a large amount of small prey items that you could eat. In this case, there won't be small fish, but there will definitely be things like amphibian larvae in it. And so as your size changes, your own probability of becoming prey for something else changes, and that may then influence what type of environment you live in, and what type of environment you live in affects the diet that you eat, because that affects what is available. Going back to that initial slide where we talked about the things that influence what your diet is, one of that is resource availability. Resource availability is influenced by where you find yourself living. There's also variation in diet over time. There's seasonal variation. This is a figure from your book looking at annual variation. And once again, this is probably not reflective of these animals choosing these different groups, but these are probably just a result of year-to-year variation in the availability of anurans versus leeches versus fish. Looks like when they can get anurans, they will gladly feed on them. And these are probably years when water breeding was low in anurans, and as a result, activity was low in anurans, and so they shifted to more readily available prey items. We don't normally think, or we haven't traditionally thought of amphibians, reptiles as being capable of learning, but there are a number of papers that are coming out now, and more graduate students in graduate programs are becoming interested in this over time, in this idea that they're capable of learning. So every spring, at the beginning of spring in January and February and into March typically, I review grant proposals from the Herpetologist League that are from graduate students trying to get small grants to fund their graduate work. And over the years that I've been doing this, there have been a number of graduate students who have been interested in studying learning in particular groups of lizards. And so organisms can learn about their prey. And in this case, they put a particular species of skink in a maze, and the maze basically took you to a point where there were prey, and they looked at how long it took the individuals to learn how to navigate the maze to get to the prey item. And they actually found that the incubation temperature that the individual was incubated at as an embryo influenced their ability to learn after they got out, after they emerged, after they were born. And so the success rate of warm incubated babies versus cold incubated babies was higher. They learned more after fewer numbers of trials, and the time that it took them to find the insect prey was lower in the warm incubated individuals. So individuals are capable of learning, and there are environmental factors that affect learning in these groups. This is different than an innate response to prey. You probably don't recall this from your first year, but when we were talking about prey specialization in garter snakes, I threw these two pictures up on the screen. This is a baby garter snake, a neonate garter snake that has just been born, and on this swab has been rubbed on either a salamander or a frog or a slug. And when you present these scented cotton swabs to a snake, the snake flicks its tongue to gather scent particles off of the swab, and that's how this thing goes about smelling. We'll study the vomeronasal organ here in a few minutes. Well, as it turns out, if a snake is interested in that scent, the tongue flick rate goes way up. So rather than flicking the tongue out slowly, it will start to flick the tongue out a lot. And so you can measure the tongue flick rate, and that turns out to be a measure of whether or not the animal is interested in that particular scent. So you can present different scents on a cotton swab and measure what their response is. And in this particular species of garter snake, there are coastal populations and there are inland populations. And Steve Arnold was interested in seeing whether or not they had preferences for certain scents. And the coastal populations tended to have high preferences for slug scents, and the inland populations tended to have not very high preferences for slug scents. They were more attuned to amphibian scents. And if you took inland snakes and bred them with coastal snakes, what you found was a shift in their preferences, such that the F1 hybrids from this had a more even distribution of preferences, actually skewed more towards the inland population. And similar thing was true, although more pronounced, for the North Coast versus crossed with inland populations. So there is evidently a genetic component for what the prey preference is of these garter snakes, depending on what environment that they find themselves in. This would be an innate response to prey as opposed to a learned response to prey. And species can have both. It's not an either or sort of situation, but just recognize that both are possible. And this actually brings us to the vomeronasal organ, which we'll probably end here today. The vomeronasal organ is the organ in the roof of the mouth. It's basically an opening in the roof of the mouth such that the olfactory epithelium that we normally think of as being separated from the mouth by a hard or soft palate. So if you feel with your tongue up into the roof of your mouth, you can feel that there's a bony part of your palate, and if you feel further back, you can feel where the bony palate ends and where the soft palate begins further back. Well, that basically is just a shelf of bone and connective tissue that separates your oral cavity from your nasal cavity. In snakes and some lizards, there is an opening that connects those two cavities. And what will happen is the snake will use its tongue, it will extend its tongue out, it will collect molecules from the air or from surfaces, and then it uses the forked tongue to transfer those molecules to the nasal epithelium directly, and that gives it a much more sensitive sense of smell than if it was just breathing these particles in and then the particles pass over the nasal epithelium in an air column. And so snakes are well known for having a vomeronasal organ and using this for a variety of reasons, one of which is tracking prey. But another function of this is to track mates during mating season. And so you will see male snakes following the scent trails of female snakes. I think I mentioned to you earlier in the semester, back in 2006, I was riding on the Katy Trail and a copperhead was in the middle of the trail. And so I pulled over and I took a picture of it because it was a copperhead. And I took pictures of it until it kind of slithered off of the trail. And then I was putting my my camera away and putting it back in my pack. So imagine this is the Katy Trail. I came upon a snake. She was heading this direction. I took some pictures of her. She continues to slither off in this direction. And then as I'm putting my pack back on my bike, or putting my camera back on my bike, another snake pokes its head out onto the trail and then continues to slither across the trail right where the female had been. So this first snake was a female that is in breeding condition because this was in fall of the year, it was September. And then a male came along right behind her following her pheromone trail and following it fairly precisely. And so this particular heightened sense of smell is very useful to snakes. I mentioned prey trailing behavior earlier in the semester, where you can create a maze where you basically lay down a scent trail or fail to lay down a scent trail, and you see the degree to which a snake can follow that scent trail. This was done in rattlesnakes from the desert Southwest by David Chazar, the guy that I learned these techniques from. And what they did was they, in some cases, allowed the snake to strike the prey item versus not striking the prey item. And then they either left a mouse trail, or they failed to leave a mouse trail in this arena. And they could tell whether or not the snake was trailing because they basically did it in a big box with paper on the ground. The snake has an opening here, and then they drew a thick line that kind of meanders around the arena to go to a rock, and behind that rock is where the mouse is the dead mouse is laying. And then they drew center lines to either side of this, equidistant, to basically create a trail. And then they measured how often the snake was within this trail. And so the amount of time that it took you to intersect the heavy line, actually come out of the place where you're hidden and get on the trail, it took them longer to do that when they didn't strike the prey item. The percent of the time available that the head was between the fine lines, they were doing a much better job of trailing the animal after they had struck it, and when there was a trail to follow. Where there was no trail to follow, they just kind of wandered around looking for it. The number of seconds the head is between the fine lines, once again, when there was a trail to follow, they stayed more on the trail than when there was no trail to follow, but both groups did better when they had struck the prey item. The number of seconds required to discover the mouse was much longer in situations where they didn't have contact with the prey item. When we look at the percentage of total time that the head was between the lines, once again, much stronger where there was a trail to be followed and the animal had struck the prey item. And then the percentage of discoveries within 20 minutes, you're much more likely to discover the food if there's a trail to follow, but also if you had experience striking the animal. So being able to follow a trail to prey after you've struck it and envenomated it, and then it's run off someplace to die, really increases your foraging success because you have the ability to trail prey because you have a heightened sense of smell due to something like the vomeronasal organ. And at that, we'll cut off there and we'll pick up at foraging modes and continue through interspecific interactions on Monday. Questions?
Oh, I do have a question, but
Yeah.
it's not about today's lecture.
Okay.
So our collections that are supposed to be due next Thursday.
Yes, they're not going to be due next Thursday because we did not go out and collect frogs yesterday. So on Thursday, what we will do is we will go out and collect frogs. And then that will probably be the last of the thing because I have your photos. I just haven't uploaded them yet. I need to get that done over the weekend. So you have photos of things that we've taken when we've gone out. Next week, we will collect some frogs. After we've collected the frogs, we'll come back in the lab and we'll preserve them. Take them a couple of days to harden up. And so we'll just have that collection be due during finals week. But it will basically be mostly photographs and one preserved specimen from each of you.
What about the turtles?
Oh yeah, turtles. So when we go out next week, we're going to be passing some ponds. And those ponds that we pass are going to be among your best bets for getting photos of turtles. And so on the old golf course, which I've gotten permission for us to go and hang out at, the college is aware that we're going to be there. The gates are locked, but you can walk around the gate. So you'll see where the location is and you can go back and visit. The way to get turtles is to either trap them in some way, or you can also stop on a highway. If you see a turtle crossing the road, you can stop and get a picture of it as well. But one of the best ways to do is going to be to just hunker down at a pond and use a long lens. So in the camera setup that we have a 150 to 500 millimeter lens that is zoomable. And you can zoom right in on a turtle so that you can be some distance from the turtle. And if it's out on a log basking in a pond, you can pretty easily snap a picture of a turtle. And there are a number of ponds at the golf course. One of them in particular has a number of logs sticking out of it. And you can, on any given day, you can find turtles hanging out on those logs basking. And so
What
we'll
do you
pass
think
those
bets are
next
that
week
I could find a lizard?
What's that?
So what do you think the bets are that I could find a lizard next week?
Lizards are going to be harder because we're going to be in a pond environment. But once again, I would just go back out to Blue River Glade if you want, or you could also go out to Martha Lafitte Thompson, where skinks are pretty common. Another place where you can go to find lizards, and once again, you'll probably want to bring the long lens with you. So you guys can check. You guys can borrow the camera setup from me anytime you want. Just tell me that you need it, grab it, and go. Let's see. Let me pull up Google Earth. Can you guys still see my screen?
Yeah.
Okay. One place that it's pretty easy to find collared lizards. They don't normally occur in this part of the state, but there's an introduced population of collared lizards not far from here. And let me get Google Earth up and I will show you exactly where you can find them. And you might want to bring a pair of binoculars. I have a pair of binoculars that you can borrow. And once again, you're going to want to bring the long lens for this because you might be able to catch them by noosing them, but you might not. So we're at William Jewell College up here. If you go down, 291, cross the river, keep going south to Blue Springs, cut over off of I-470, there are these two lakes right here. The northernmost lake is, I believe, Blue Springs Lake. Yep, Blue Springs Lake. Stop zooming in. I don't want you to zoom in anymore. And so if you come down 470, so 291 turns into 470, 470 turns into Highway 40. And then you can take any of the entrances into, gosh, what is the name of this park? There's this big park that surrounds both of these lakes, and I forget its name at the moment. You can come in here, and right along the dam here, this dam is basically a gigantic boulder field. And if you walk along this little road where this boulder field is, and if you scan the boulders up here, you will occasionally see colored lizard heads sticking up or entire colored lizards basking out on rocks. And then you can set up the long lens and get a photo of a lizard from that standpoint. Also, right next to this dam is the quarry where they presumably got all the rock that went into this dam. And along the edges of this quarry, especially on this side, there are a bunch of exposed rocks. If you go and flip the rocks along in this quarry, you will find a lot of snakes, and you will also find occasional skinks underneath those rocks. In this way, it's kind of very similar to the environment at Blue River Glade, except that rather than all that rock being exposed naturally, this was exposed by quarry operations. So that's another place to go and get lizards. So reliable spots would be Blue River Glades, which you've already seen that there are a number of lizards at Blue River Glades. There are colored lizards here at the dam and skinks over here at the dam as well, at the quarry. And then, of course, Martha Lafitte Thompson. If you flip some logs and stuff like that, you will occasionally find skinks under those as well. So there's a number of places where you can go and get lizards pretty reliably. So yeah, so next week in lab, rather than just turning things in, we'll actually go out to the field and collect some frogs. Otro más. All right, then I will see you guys on Monday.
Paul, can I talk to you for a second?
Yeah.
Did you see my email by any chance?
When did you send it?
It was about